we move first with Dr. Michael Orth talk, and then we will move forward with Professor Julie Stout. Thank you so much.
So good afternoon, everyone, and thank you, Saul, for the kind introduction. Thank you to the organizers for inviting me to speak today on the visual system in Huntington's disease. I have, for that purpose, so I can see my own presentation, put on my glasses, because if I hadn't, I would probably have the visual acuity of a goldfish or a rat, and I wouldn't be able to see clearly. Just to illustrate how important it is that you see clearly, humans are a visual species, but more so is the eagle. The eagle has about 4 to 8 times higher visual acuity, meaning he can see things clearly from about 5 times as far away as we can. This means in flight or sitting perched on his tree, he can see a small animal from about 2 to 3 kilometers away. Eagles are not known to suffer from nearsightedness or farsightedness. It wouldn't be good for them to be not eagle-eyed, because that would be a severe disadvantage and probably they might starve to death. However, the world is not populated by eagles, but is rather overpopulated by people. It means visual acuity alone is probably not what makes the difference. What makes the difference is probably what happens with the primary information that reaches the brain through the eye, the optic nerve, the optic tract, and then ends up in the primary visual cortex. Just to go through a simplistic diagram of what happens with this information, I will show you in a minute, because I've got ahead of myself as a little diagram. Just to remind you, vision, as we know, is a key sensory modality, and really what we want to derive from the information is the what, the where, and the how of visual object recognition. It's also important to know how these objects move and change with regard to how we behave, in particular when it comes to social perception. Now, the visual information that enters the primary visual cortex helps us to make out shapes, colors, and other aspects of objects, which then lend themselves to a proper and in-depth assessment, hopefully triggering some visual contextual memory. This, together with information from other modalities such as sound, smell, or taste, can help us to recognize what we are seeing and then give meaning to what we are seeing. Meaning means what it means in that very instant to us and how it will influence our behavior. This is an example for how we immediately perceive. These are all similarly looking, but also quite differently looking shapes, but we immediately see they're all glasses from which we can drink, except one object that is not so dissimilar from the others, but we can make out this is probably a glass bowl and not a glass from which to drink. Now, there's more information that we then perceive that helps us to make out different things on a different level. Now, these are all glasses, but there's something colorful and nice looking in them, and there's some nice embellishments like some orange zest and straws and ice cubes and what have you to it. So this helps us to recognize what we're looking at is glasses, but really what it means is these glasses are full of something that we call cocktails. Now, this is a similar level that we are looking at here, but you can make out immediately that at the bottom row, the left 3 drinks are probably not cocktails, unless somebody would like to call a beer a cocktail. Particularly important is the recognition of faces for us. Now we can immediately see these objects are what we call faces. These faces belong to people of different ages. The 2 sexes you can make out immediately. You can make out children, older people. Some of them bear a friendly smile, others have quite a neutral expression. facial recognition is particularly well trained, so that you can even, in this artistic rendering of Marilyn Monroe's face by Andy Warhol, make out that this is a face, belongs to a person who's female. We know that she's famous, and hadn't I told you it was Marilyn Monroe, most of you would probably have known this is Marilyn Monroe, even though a lot of the information that we normally perceive from a face in everyday life are not present in these images. Attention plays a very important role in what we see, because we are bombarded with a lot of visual information all of the time. So attention helps us to focus on what we think we should be looking at now. So if I tell you on the left-hand side in this illusion to look out for the beak and the head of a duck, you would probably see it. If I told you to look out for the ears and the head of a hare or a rabbit, you would then see the rabbit. On the right is another example for an illusion. It depends what you're looking at, what you're looking for, whether you perceive parts of a face from the front or from the side. Attention has to be focused on what we think is most relevant to our behavioral goals. Sometimes these behavioral goals, or what we perceive as relevant, may not be as relevant for ourselves, or may not be in our best interest, let alone in the best interest of somebody else in that scene. So that woman in the car that's about to be hit is probably not so happy with what the girl on the phone is currently paying attention to. Particularly important is the perception of movement, because what you've seen so far are stills. Movement is something that's part of everyday life. Most of what moves is alive, and I wouldn't say most of what doesn't move is dead, but you know what I mean. This is an example from tennis. The tennis player receiving the ball, the player in the blue shirt in the bottom, looks very carefully how the other player starts to serve. He will recognize by very subtle changes in posture and movement where he's likely to serve to. When he can anticipate where the server is going to place the ball, he can adapt his position for the return, and this gives him a great advantage for the next shot. It's very important to constantly assess the situation, because you see how the ball flies, you have to adopt a different position for hitting the ball optimally, so to give it the desired direction. These assessments are also extremely important when it comes to social interactions. If we're in a group or we're busy with other people, we constantly look out for how they move, where they turn their eyes to, what facial expression they adopt, how language and body language go together and where they don't. And this makes social interactions possible. Any misperception of these these social properties of facial expressions and so on can lead to difficulties in everyday life. There are three pathways that scientists distinguish. There's a ventral pathway that computes the identity of visual objects, as in the examples before, to make out are these glasses, cocktails, faces, animals, whatever it might be. We need to make an assessment of where in space these objects are located, which is the property of the remit of the dorsal pathway. And we also need to assess where these objects are in relation to what's happening. So what are we doing? And then there's a third pathway that serves these higher socio-cognitive functions. So these functions are based and work best if we can dynamically assess social cues, as I said before, facial expressions, eye gaze, discrimination, understand speech, but also see how people behave while they talk. And immediately we are able, in most instances, to interpret the actions and behaviors of others. In Huntington's disease, we don't really have much evidence to suggest there is a problem with visual acuity or visual fields. So in a simplified way, I think we can assume the information transfer to the primary visual cortex is probably intact. We got data in the prodromal phase from visually evoked potentials, which have normal latencies to indicate that the speed of information transfer to the primary visual cortex is also normal. And there's some evidence from neuropathology to indicate there's probably not much of a loss, at least early in the disease, in the primary visual cortex of neurons. There's a whole host of studies that have looked into behavioral data, so what I call recognition. I can't read this, even though I put my glasses on, and it's not the purpose of these slides for you to read them. It just shows there are loads of studies that have looked at this. They have employed a whole host of different tools and measurements, have included cohorts of different sizes, of different clinical properties, near onset, after onset, further away from onset, later stages. But in a provocative way, and Sol encouraged me to be a bit provocative, I would come to the conclusion, up for debate, that many abnormalities have been reported, most of them in manifest HD. Many different instruments were used to do so, so comparisons and the reproducibility of any of these findings is difficult. Visual cognition has hardly ever been the main focus of these studies, so they've used data from studies that were designed with other scientific questions in mind. And very importantly, it's probably no secret to say, any assessments of visual cognition have the confounds of motor abnormalities, the general slowness of information processing, the dependence on oculomotor function and attention. So if one sees an abnormality or a difference in measures on scales that depend on a number of these properties, one can't be sure why there is a difference. There's no doubt there are structural abnormalities in HD. So neuropathology documents a loss of parameter neurons in the secondary visual cortex, while there's no difference probably in the primary visual cortex. There's also very good evidence from imaging studies to suggest the gray matter has changed, including in the occipital lobe, as people move towards manifesting signs of the illness from the further away pre-HD group here in track HD to nearer onset in pre-HDB to early HD. The cortex, including in the occipital lobe, becomes thinner. The same is true for white matter. White matter is also, it's been known for many years, the white matter is affected macrostructurally. It is also affected microstructurally, including diffusivity properties of visual tracts, where, and especially in early HD patients, there are differences to pre-HD and controls. There are functional abnormalities that have been reported in HD. There are different networks of visual function that can be made out, one in red and one in blue on the left. And there's even some evidence that there's less left fusiform gyrus activity in HD. But it's important to always consider that resting state fMRI or PET operates in a very poorly controlled environment, and the relevance for any of the behavior that needs visual cognition is unclear. The selection of tasks for functional studies is in this context extremely important, and tasks should avoid any need to press buttons or to indicate choices with speaking. So ideally one should use videos or other things that are used in visual cognition research. There's no doubt there's a great deal of practical relevance of visual cognition. One example from social cognition is, can I trust another person? This is extremely important. Well, when we lived in a less protected environment, it was very important for survival, but it's still very important. And we make this assessment in a split second. Driving is another prime example for how relevant visual cognition is, because driving always concerns safety for the driver and anyone else who takes part in traffic. It is also a key requirement for movement and balance as everyone can attest to who's bumped into objects while groping for the light switch at night. Rainer Sprengelmeier looked at the question of social cognition in HD, and what he was interested in is basic emotional expressions of faces, but above all, he was interested in what conclusions participants arrive at when it comes to trustworthiness and dominance. So there was a clear difference in the ratings of trustworthiness and dominance between patients and controls, with the patients doing less well. And there was no difference in a similarly complex judgment of the faces, sexes that they have seen. Patients were impaired in the recognition of basal emotions from facial expressions. And there were two groups that he distinguished, one that was impaired in the ratings of trustworthiness and dominance, and another that wasn't. Even the unimpaired group had some deficits in the recognition of basal emotions, but they were still able to rate to synthesize the information that derived from the faces correctly. So it probably means that the perception of these basal emotions has to drop below a certain threshold for the synthesis decisions become affected. Milou Jacobs looked at how patients and controls performed in a driving simulator. So one of her outcomes was how well people were able to hold a line with their cars. And she noted in manifest to patients that there was a correlation of how much they veered to the left or the right with how much they were impaired motorically or cognitively. Another indicator of a change in driving performance in the simulator was driving speed. Manifest HD patients were generally slower, but they also seemed to be doing less well on the SDMT, which is one measure of visual motor processing speed. So it might just be that they were generally slower, and this is why they drove more slowly. Assessing driving ability is an important issue, including an aging, neurodegeneration such as Huntington's disease. It's a complex function that links executive function, attention, visual cognition, speed of information processing, as well as oculomotor function. EHDN awarded the seed fund grant to Riens Huitema and Barry Kramer from Groningen. They hope to build on what Milou Jacobs has already done and include in their assessments on-road driving. And we also have founded an EHDN driving task force. So anyone who has an interest and wants to know what's going on there or join in, please have a look at the EHDN website. So to sum it up, for visual cognition research, contrary to, say, studies in stroke or when you inflict temporary lesions experimentally, it may have to be different in a neurodegenerative condition where many different systems are affected, not just a visual system. Many studies haven't really focused on questions regarding visual cognition, and it's clearly challenging to select the appropriate neuropsychological measure. I mentioned before, many tools and assessments have a motor component. They require eye movement control for tracking. They may have a language component. Lots of cognition, almost all of it is dependent on attention. And if you're just getting slower, you're also not performing quite as well on any of these tools. So to sum up what we know from the data, there's no doubt that the occipital lobe is structurally affected early in HD, and that's backed up by imaging studies and neuropathology, where the neurons are probably mainly lost in the association cortex. Behavioral consequences are likely, but no clear picture seems to be emerging. It may well be that the questions weren't chosen for the research targeted to visual cognition. It may also be that we don't see it clearly because we're using the wrong instruments. Given the relevance of visual cognition for human behavior, and that includes behavior such as driving in HD, research in this regard is clearly very important. So we need good studies with solid questions addressing visual cognition. Thank you.